So I will start to present myself as a chairman, but also I will have two presentations. The first one it's about the drip irrigation's innovation efficiency and the biofilm dynamics using regulated wastewater and drip irrigations in the field scale. So my name is Mouheb. I am director of research in INRAE Institute in Montpellier, a laboratory Géo in France. So this work was during the PhD of Vincent Moulia in collaboration between the laboratory LBE, l'IEM, and of course my laboratory Géo. How it works? So, like this. Right here. No. Okay, thank you. So just to start, I would like to present you the general principle of drip irrigation. So the drip irrigations is the water applications above or below the soil using the surface or subsurface drip irrigations. A dripper is a really millifluidic system, can help to generate the pressure drop using the really complex labyrinth channel, depending on the drippers, type of drippers, but also using really small channel, around 1 millimeters, in order to produce the flow rate between 0.3 to 6 liters, but in associate we can use until 8 liters per hour. So the advantages of use of drip irrigations is comparing to other irrigation technologies is to save directly the water. So the efficiency of the water uses is higher than, for example, the sprinkler irrigations. And what is interesting for the reclaimed wastewater, you reduce the contact between the water and crops. So we can consider generally, according to also the water use irrigations, is the safer technology comparing to considering the sanitary risks. But the disadvantage of this technology of the irrigations is really the effect of the small section channel for the flow. So in these drippers, you can have a risk of the clogging, physical, chemical, but or using the reclaimed wastewater, you can have also development of the biofilm and the clogging of the drippers in time. But there is also a question of the cost. So this technology is generally considered higher, is more expensive than flood irrigations, for example. Next one, please. So in these experiments, with the aim of the objective of this project was to better understanding the biofouling of the drippers, considering the real field conditions during the summer period of irrigations in the Mediterranean climate context. And the sub-objectives of this work was to consider not just the small length of the pipe, but really long pipe in order to consider the flow behaviors along the pipe, considering different type of commercial drippers, but also optical drippers, but also considering also different flow rates and flow regime from turbulence in the upstream to the downstream, how it can affect the biofouling and the biofilm growth. And using the different drippers, we monitored also the flow rate, and optically we quantified the biofouling inside the drippers. And in order to answer how this biofilm can affect the water quality, we also analyzed opportunistic pathogens and E. coli indicator in the biofilm, but also in the outlet water of the drippers. Next one, please. And just to mention how comparing to the lab conditions, the field will affect. You will have the effect of the real environmental conditions. And here what is important is to consider the variability of the water quality, but also the effect of the temperature during the no irrigation period. Next one, please. And for that, we carried out this experiment in the field, in our platform in Murviel and Montpellier, and using the reclaimed wastewater treated by the constructed wetland. And in order to boost or to enhance the development of the biofouling in our system, we irrigated 6 times per day during 13 minutes, and the duration of these experiments was around 4 months. Please. And for that, we used the different methodology, complementary methodologies. So at first, we monitored the biofouling with quantifications of dry organic matters in the pipe and in the middle and the end of our experiment. And inside the drippers, we have the possibility to quantify using optical coherence tomography the biofouling. And we analyzed also 2 opportunistic pathogens, Legionella pneumophilia and Aeromonas hydrophilia, in collaboration with the laboratory LBE, and as an indicator of fecal contaminations, the E. coli. Next one. So for our experiment, we monitored the flow rate and also the biofouling in 6 lines using commercial drippers with 0.6 and 1.5 liter per hour, but also we used this kind of experimental optical drippers in order to monitor in time the development of the biofilm using the OCT optical methodology. So it's important to consider the difference of the flow rate and the flow regime and shear stress in our pipe, how it can affect the biofilm. It's important to know in the beginning and the upstream of our pipe, we have turbulent regimes with high shear stress, but in the end of the line, you will have a 0 flow rate because you will irrigate, you will have the output of the drippers, and then you will have a laminar regime and low shear stress. So we will see how it can affect our biofouling and bacterial communities. Next one, please. And yes, first results to consider in the field case, in the field scale, is the effect of the temperature. Here we monitored the temperature in the inlet and outlet of our pipe. And here in this figure, you see the variations of the temperature, which can reach 50 degrees. Water quality, the water temperature can reach 50 degrees in the pipe during the summer. Next one, please. So this temperature will affect, of course, the biofouling in the pipe. So we have the effect of the positions. The first results, it's about the dry matter, biofilm dry matters. So we observed the higher quantity of the biofilm in the downstream of the pipe comparing to the upstream, and higher concentrations of and percentage of organic matter also in the downstream, due to the conditions, flow conditions, laminar conditions, and low shear stress in the downstream of the pipe. But comparing to the previous results we obtained in the laboratory condition with the ambient temperature between 20 degrees, we observed slower biofilm growth due to the high temperature of the water during the irrigation period. Next one, please. And this condition will affect also the bacterial concentrations. So we observed also, if you see in this histogram, high concentrations of the bacteria also in the downstream, due to to the flow conditions and the shear stress in the downstream of the pipe. Next one, please. Now, how it after the pipe, we will show you some results about the drippers. Here, dripper flow rate and our observations of the commercial drippers. So using this both and during 4 months, both drippers and during the 4 months of experiments, we didn't observe degrees of the flow rate comparing to the initial measurement of the flow rate. And when we opened the drippers using our microscope, we didn't observe the biofouling of the drippers, but just planktonic particles due to detachment of the biofilm in the pipe, which explain the no effect on the drippers flow rate. But using next one, please. But using our optical drippers, because we have this transparent pipe, we observe the degrees, as you can see here, of the flow rate in time. Degrees lower than 75% of the initial measurement, and the development of the biofilm volume can exceed, will not exceed, but it's around 20% of the volume of the total flow sections. So it's just after 4 months, we can suppose after 1 year or more of irrigation with reclaimed water, we will have more clog in our system. Now concerning the next one, please. Now concerning the bacteria diversity in the pipe and the drippers. So the first research showed no significant difference between the sampling zone. There is no effect between upstream and downstream of the pipe, but there is an increase in time of the bacterial concentrations, but it's with the abundance of thermophilic bacteria due to the effect of the field conditions and the high temperature of the water during no irrigation period. Concerning the opportunistic pathogens, we didn't have a possibility to quantify these two opportunistic pathogens because it was lower than the quantification threshold. But what is important is the concentrations of E. coli in the biofilm we observed and we measured higher than 2 log copy per centimeter square. And there is no influence of hydrodynamic condition on the concentrations of opportunistic pathogen in the biofilm. Next, please, and conclude. I don't know if I still have time, but so take home message. The first one, there is no reductions of irrigation efficiency detected using our drippers, commercial drippers, during the short period of irrigation, 4 months. But what is important is to consider in the field scale comparing to the lab scale the effect of environmental condition, which have more effect than the hydrodynamic conditions. And what is positive, we didn't have a possibility to analyze or to have the concentrations of opportunistic pathogens in the biofilm, but we have the effect of the biofilm detachment on our bacterial communities analyzed in the water, in the outlet of the drippers, and some concentrations of the E. coli, which can pose the question of the effect of the barriers due to drip irrigations, which is not already considered for the monitoring in the applications of the regulations in the field. Last. And so for more details, we already published the part of this work in this paper, so I encourage you to read this paper, and thank you for attention. One question, maybe? No question. Next presentation, please. Still me. Now, next. Okay, I will start. So unfortunately, the PhD student Carmelo Jean-Louis was not able to come for the conference, so I will present his PhD project about the multi-compartment mass balance model for E. coli and the spore dynamic in treated wastewater reservoir. This project is in collaboration between my laboratory Jérôme Armand from LBE, but also Mistea and the BRL exploitation company. Next one, please. So this work is not dedicated to the drip irrigation, but to important component in the irrigations is the reservoir. So the storage of treated wastewater is an important component to maintain and to manage the water and enhance the availability of water during the irrigation period. But the storage during the time can affect the different physical, chemical, and biological and pathogen parameters depending on different depending on the input water quality, climatic conditions, but also external contributions. But it's important also to consider operationally this reservoir in order to implement the regulations and water use regulations and monitoring the different parameters, pathogen parameters, E. coli, spores, and bacteriophages. So for that, during we consider in our reservoir there is many parameters can affect the water quality, as you can see in this scheme. So the UV radiation, the temperature, but also the inlet concentrations will affect in time the reservoir water quality. And there is also other mechanisms, please the next one, can affect the water quality in time. Next one is the difference of temperature between the outside and the water temperature, which induce the stratifications and will change the mass transfer of the pathogen. And you have also the effect of the flow behaviors inside the reservoir, depending on the geometry, the depth, and the ratio between surface and depth of the reservoir. Next one, please. And so during the PhD of Carmelo Jean-Louis, so our objective and main objective was to understand the spatially and topically the evolutions of the and dynamic of the evolutions of the and pathogen indicators. The three pathogen indicators of the regulations is which we considered the E. coli, bacteria, spores, and the bacteriophages in open air reservoir filled by the treated wastewater, taking account not just the irrigation period, but also the long-term storage during the winter season without the irrigations, and then during the irrigation period. So the idea it was also to develop a dynamic model in order to represent the main mechanism associated to the variations of the pathogen indicators in order to formulate some strategy can apply for the multi-resources reservoir. Here we consider the multi-resources reservoir management is the mix between different water qualities as the conventional water with treated reclaimed wastewater. So for that, we need to develop a specific model in order to think about the different scenario for the management of this mix and blending of this water in the reservoir. Next, please. For that, we carry out our studies in two case studies. The first one in Mouheb and Montpellier platform, and the reservoir filled by the treated wastewater from the constructed wetland, and with high ratio between the surface and the depth. And the second case study was in Roquefort-les-Corbières with treated wastewater from the activated sludge plant with lower surface depth. So we have a depth of 5 meters in the case of Roquefort-les-Corbières, and in the Mouheb-le-Montpellier reservoir we have a depth of less than 2 meters. And during our experiment, we sampled in different positions, depending on if horizontally and in different position in the depth of the reservoir. And for our experimental experiment and our models, we monitored different parameters, not just pathogen indicators, but also the concentrations of nitrate and ammonium and different physicochemical parameters. And we considered also the climatic variables and also the hydrodynamic variables. Next one, please. And for the model, we considered 2 compartments, 2 perfectly mixed reactors. And in our model, we considered the water quantity balance equations and the mass balance equations, taking account sources term and loss term. Please, next one. So for the first result about the E. coli variations during the no irrigation period, so during the long-term storage, we observed after the filling the reservoir linear and uniform reductions of 3 log of E. coli concentrations after the 1 month, which is interesting to consider in this case the reservoir as an additional barrier for the pathogen degrees. So, but during the irrigation period, we have variations of E. coli concentrations according to the refilling and irrigation cycles. We observed sometimes some difference between the top and the bottom of the reservoir due to the stratifications, thermal stratifications. Next one, please. But concerning the second indicator, the porous concentrations, in this case, there is no significant effect of the storage period in the no long term or during the irrigation period, just the effect of sedimentation during the no irrigation period. Next one, please. So for our model, E. coli model, we can see here our result of the modeling of the E. coli variations represented here in the blue and red. There is reliable representations comparing to the measured data in the case of the long-term storage or during the irrigation period. Please, next one. And the same result about the spores modeling. So we observed also our result fit well the result, the experimental result, considering also a different compartment between the top and the bottom of the reservoir. So now from this result, our idea is now to test different scenario of the multi-resources mix. So here is the first result. Considering the scenario of the storage with just conventional water, so here you have the first result. Here after we start to fill the reservoir with the conventional water, we have 2 log, and during the time you have a degrees of the E. coli pathogen concentrations. But when we start to introduce some treated wastewater in the reservoir, we have the increasing of the of concentrations, which now will help us to maybe to think about the strategy in order to consider different percentage of treated wastewater mixed with conventional water, or the position of the input and during the input of the reclaimed wastewater during the time. So just to conclude, next one, please. So the long-term storage can reduce the until 2 and 3 log the E. coli concentrations, but during the irrigation period, unfortunately, we have the increasing of and the resultant time is not enough to reduce the concentrations of E. coli. Concerning the spore bacteria, we also observed this high value persistent of this bacteria, all two sedimentation phenomena observed during the winter. The E. coli and spores dynamic using our model can reproduce it correctly, the measured data by a 2-compartment model. And now we are taking to apply this model for multi-resources and the mix of resources in order to take the best and to select the best proportional of conventional water with treated wastewater in the reservoir, considering the regulations, water use regulations. To conclude, thank you for your attention, and we published partly this result in this paper in environmental technology, our experimental data, so I'll encourage you to go more details. Thank you.